all the stuff that circulates in the blood have an effect in different pathologies, but also it changes as we get older, not in everybody, but in most of the population.
Welcome back to Longevity Roadmap. This is Buck Joffrey, and today I'm really psyched about sharing this interview with you guys. It is with really accomplished scientist from the Buck Institute, Dr. David Furman. And we've talked on this show so much about the role of inflammation, called it inflammaging, and as part of this whole process of aging and the development of chronic disease and so on. So this is a really fascinating interview because Dr. Furman is really sort of at the cutting edge of not only figuring out what is behind inflammaging, but what kinds of things we can do to intervene. He talks a little bit about some tests that we can do that are commercially available, which probably are better than using epigenetic clocks, frankly, and also alludes to some studies that really suggest that we might be on the precipice of actually being able to reverse some of the stuff, which is huge in my view. I've come to believe that the immune system and inflammaging is fundamentally probably the biggest factor, at least on the clinical side, of aging, and is going to really affect not only lifespan, but also health span as well. So if we can get our, hands around this and start actually reversing some of the inflammaging effects that we naturally get, it's huge. So anyway, I really enjoyed this interview, and we will have it for you right after these messages.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to the show, everyone. Today, my guest is Dr. David Furman, one of the world's foremost experts on immune aging. He serves as chief of the AI platform at the Buck Institute for Research on Aging and directs the Stanford 1000 Immunomes Project. He holds a PhD in immunology and trained at both the Pasteur Institute in Paris and Stanford. His work has reshaped how we understand the age immune system and how we might actually reverse it. He's a true leader in the field, and it's a privilege to have him on the show. Welcome, David.
Thank you so much. Thank you for having me on the show.
So why don't we start with this? We talk on this show, the audience has been exposed to this concept of inflammaging. Would you, though, mechanistically, if you would, what's happening in the immune system as we age, and why do you believe immune dysfunction is such a central driver of age-related disease?
There are major changes in the immune system. The immune system has blood as a pipeline that connects different organs. And in the blood, there are cells and there's liquid, right? The serum or plasma. And there's proteins that interact with one another and that talk to cells with this pipeline that I'm alluding to. And so there's a variety of different changes, including shifts in the frequency of cells and increases in, say, memory cells, the CD8, the CD4, the other NK cells and stuff like that. And there's also shifts in the naive T cell pool, which is quite critical for the responses to, say, vaccination or infectious diseases. There's also huge amount of changes at the level of proteome, metabolome, all the stuff that circulates in the blood have an effect in different pathologies, but also it changes as we get older, not in everybody, but in most of the population.
Yeah. So I guess the question I have in part is, what is causing this shift in the immune system as we age? I mean, we recently spoke to, for example, one thought, Dr. Greg Fahey, who is really interested in how the thymus plays a role in immune aging, and has talked a little bit about the degeneration of the thymus and potentially reversing some of the immune function of aging by restoring the thymus in the trim protocol. What are your beliefs in terms of, the underlying cause of some of these shifts?
The changes that occur at the level of the immune system are relatively, not very clear and understood, in that the origin may come from different sources. So decreasing thymus size, the involution of the thymus, is believed to contribute to a loss of a naive T cell pool, and that definitely happens. But also you see changes at the level of the bone marrow. And also you see changes in the homeostatic proliferation mechanisms that happen in the periphery. And so it's much more complex than we understand. I like to say that the immune system is really a sensory system. And so, much like your sight or your hearing capacity or your taste, right? It captures information from your environment. It processes that information and has an output. And so, in the case of the immune system, the major difference with other more classical sensory systems is that the information doesn't become conscious, but you have an output, and that output is inflammation. So I like to get to that because I believe the more you have inflammation in the body, the higher the risk of developing many of the diseases that we know happen with aging, including a loss of the ability of our immune system to respond to pathogens. And one of the causes that we believe are root in that mechanistic process is that we're exposed to so many things in our environment. And so, say, every jog, every meal, right? Your happy microbiome, will increase the anti-inflammatory molecules. A poor microbiome will decrease that. And so you're responding to your environment. And because we're not, in a very healthy environment, but rather quite toxic environment, our immune system deteriorates, and we have increases in inflammation.
So the concept, again, just so I understand, is that the inflammation that we're seeing in our body is a cumulative effect of the inputs that we're getting from the environment. That's kind of what your take is, correct?
Absolutely. Imagine you're overtraining, you're going to be inflamed. Your zone 2 cardio, you're going to go and lower your levels of inflammation. There is very active and malleable change that the immune system is dictating as a rheostat for how healthy your immune or your environment is. Also, the level of your senescent cells. We know that a lot of the inflammatory molecules that circulate in our bodies come from senescent cells, and they may not only originate from the immune system, but also from endothelial cells. So it's a little bit more complicated than we think.
Tell me, so you're leading research, obviously in immune aging, but you're also chief of the AI platform over at the Buck Institute. So how are you using artificial intelligence to study aging and immune decline?
We're using this as a tool for mainly two different aspects or areas, right? One is the AI, bioinformatics, computer sciences have evolved in parallel with biotech. And with the biotechnology that we have today, we can measure hundreds of thousands of parameters from blood, from tissues. We measure the whole proteome, the whole metabolome, all of the genome, right? It would take me 100 years to be able
Yeah.
to analyze all these data if I don't apply AI machine learning to it. And so, on the one hand, it accelerates the discovery of biomarkers. It enables us to develop new clocks that indicate rates of aging in your immune system. And so it facilitates, right, the work that could take many, many years to achieve. And on the other hand, it really now with supermassive language models and generative AI, we can start assigning different genes or gene sets to hallmarks of aging, for example, and start understanding a little bit better. So the idea of these 12 hallmarks of aging, right, that we all know of, is poorly defined, and it's been largely crafted from studies in animals and in vitro cellular or models. But now with LLMs and the help of generative AI, we have identified which pathways and which gene sets are associated with each of the hallmarks of aging. So it basically gives us a better sense of the biology around hallmarks of aging that's contributing to that decline in health span.
So I believe that I believe this is part of that AI effort, but you developed this eye age clock to measure biological aging through immune system signals. Is that correct? And how does it work? I mean, is this essentially just using large amounts of information essentially to compare it with some sort of sample group with of known aged immune systems? Is that essentially how it works? Or
Right. The IH, or inflammatory age clock, is the poster child for how we use machine learning and AI in this case.
Mhm.
It's part of the Thousand Immunomes project, right? We started the Thousand Immunomes in 2007. It's been almost 17 years, 18 years. And we collected a huge amount of data. Part of that was using proteomics to understand immune system behavior, inflammation, and so on and so forth. Because inflammation is so poorly defined, and it's not the type of inflammation that happens when you cut your finger, right, and gets red and swollen and warm. Those are cardinal signs of acute inflammation. They're very well established markers. The interleukins, right, the different interferons. So those are well-established markers. Even C-reactive protein is
Mhm.
now used for pathogen detection. Inflammaging is poorly defined, and so we aim at defining this. When we build a linear model, we have very poor predictions. And so we decided to have a more sophisticated model, a non-linear model that can capture redundancy in the data, denoise, because humans are noisy by definition. And so that's the type of deep neural network that we used. It's called guided autoencoder. Forget about that jargon, but suffice to say that applying that to the Thousand Immunomes proteomics data, we were able to derive this inflammatory clock of aging that's predictive of multimorbidity, frailty, cardiovascular disease, exceptional longevity, and immune system behavior.
So, how, I mean, does it assign you a certain, I guess, immune system age? Is that kind of how it works?
Yeah, so it's really an inflammatory age score.
Inflammatory age
Yeah,
score. Yeah,
yeah,
the reason I'm asking is, there's this, the Horvath clocks, the epigenetic clocks, and all that are out there. It seems a little bit more straightforward on at least what they're trying to, they're basically comparing with a big set of chronological age people and trying to match up biological age with that. But in this is not, as you said, it's not linear. This is a little bit more complicated. So tell us, example of what kind of information is output from that IH.
yeah. So I love what you're saying, that this is not entirely compared with other clocks. I actually don't like to call this a
Yeah.
clock. It's the burden of inflammation you have in the body. And so it gives you a percentile on comparing your levels against a normal distribution of other people of the same age. So it puts, you remember those
Yeah.
growth charts of your kids,
Yeah.
right? You want to
Right.
be in the upper percentile. In this case, you want to be in the lower percentile, right? And it also gives you the top 5 proteins that contribute to this inflammatory age clock. And these are not the proteins that are the usual suspects, right? These are CXCL9, also called MIG. It's a protein that, when it's elevated in the elderly, is largely associated with cardiovascular aging. Eotaxin, or CCL11, it's another protein that, when it's elevated, you have a higher risk of developing cognitive deterioration. And then you have another 3 proteins, 2 of them participate in immune system reactions. You want them to be elevated, actually. And trail, which takes care of the apoptosis of senescent cells. And so if
Yeah.
you have it in low quantities, you have accumulation of senescent cells. So it gives you, in summary, the level of, or score, or percentile of where you're at in comparison with others of your same age, and the levels of these 5 most predictive proteins.
Uh-huh. And just for clarity, if you take a, protein like, I know CXCL9 is one of the ones I read about. How do we know that this isn't just that it's not, or maybe we don't know that it's not just a biomarker, but a causal driver of aging related decline?
Well, you have to do the experiments, right?
Yeah.
And we did so. We did it in. Unfortunately, there's one of the only models that we had at the moment was using animals, right? Today we have organoid models that you can perturb, and so you can have kind of a cause and consequence.
Yeah.
And we did it in animals. So we observed, say that CXCL9 is elevated in older mice. And when we knock it out, right, we basically knock down the gene, you have a significant change and an improvement in contractility, in immune system reactivity, in a variety of different aging metrics. And so you need to do the experiment. There's no way
Right.
around that.
So this was basically where you silenced the
Yeah, I
CL9.
apologize. I wasn't clear enough, but that's exactly what's happening.
Yeah.
We silence the gene, and then you have
And that was in endothelial cells,
that's right.
correct? Yeah.
That's exactly right.
And then essentially that reversed some age-related dysfunction of the endothelial cells. So
That's exactly right.
big implications, obviously, are there for potential cardiovascular disease. So how close are we to translating something like that into a therapeutic intervention in humans? Because that obviously seems, with cardiovascular disease, I think most of us, I don't know if it's most of us physicians, but certainly the physicians who are following, latest concepts on this, we think of cardiovascular disease being the driver of this being not only ApoB particles, cholesterol particles, but a significant portion being inflammatory. And we've been following that with, high sensitivity CRP and that kind of thing. So here, I guess this really attacks one of the major arms of cardiovascular disease. So how close are we to translating something like that into a potential therapeutic?
There are certain limitations with this kind of approach. I'm going to be super blunt here. The immune system's complicated.
Yeah, sure. Of course. Yeah.
So if you block a protein that's important for other functions of the immune system, and that's typically what happens. You don't want to block CXCR3, which is the receptor for it, because then you're dampening the entire immune response. When you get an infection, you may die. And so you have to be very selective and not block the receptor, but rather CXCL9, which is very close to CXCL10. So there's all sorts of
So you have to be very,
complexity. Exactly. And so I like to think that there are nutraceuticals that are very safe and that can lower the levels of CXCL9. We tested one in particular in a large effort for eotaxin, not for CXCL9. And we were able to decrease the levels of eotaxin, which again is critical in the development of Alzheimer's disease and other dementias.
Oh, yeah.
And simply by adding avenantramide rich beta glucans as a supplement.
Sorry,
We
what
were,
what what was that supplement?
so you have two components in soluble oat. This is basically a product that's not in the market. Collaboration with a corporate partner. It's a paper we published a few years back. It's a study that was done in 200 individuals, controls, and 200 experimental. And we were able to see as early as 2 weeks after consumption of a product that is rich in flavonoids. This is phenols that they're called avenantramide, okay, that are part of the oat, and beta glucan, 3 grams of beta glucan per day. During 2 weeks, we're able to decrease the level of eotaxin significantly. And with that, we saw a decrease in IH overall. And so that's one example that with natural extracts or compounds, we can do as efficiently, as good as with the most sophisticated drugs. So
Well, that's interesting. I'd never heard of that, and I'm surprised more of the, longevity people out there aren't all over that one because it sounds like a big one. So you were also a senior author on a recent clinical study where you basically were using therapeutic plasma exchange to see if you could reverse, quote unquote, biological age. Can you tell us about the protocol there and what you found?
sure. So the protocol is divided into 4 arms. This is a single-blinded placebo-controlled trial. The placebo was sham, and we have therapeutic plasma exchange, which basically consists on extracting a large volume of blood and sort of quote unquote filtering that blood. We dilute that blood, add albumin to it, and put it back into the patient. And so by doing so, what you're essentially doing is all of the pro-aging factors that are in the serum, you're diluting them. And so the immediate consequence of this was a significant decrease in the biological aging of these individuals as measured by over 35 different biological aging clocks. Now, the 3 groups consist of a biweekly TPE, therapeutic plasma exchange, with and without IVIG. So this is immunoglobulins administered IV, right? And then you have a monthly TPE. So there's those 3 groups plus the sham. And so the primary endpoint was to observe if we could improve biological aging. And we did, on top of that, we did multiomics, right? Which is a variety of other things that we measured in the same individuals of this study.
How are you testing biological aging there? What was the endpoint? How do you measure that?
The most, I would say, accepted way is by looking at epigenetic clocks. But I would say, there's been a lot of discussion in the space of biological aging. I have my favorite biological aging clocks are transcriptomic based, not epigenetics, because epigenetics will depend on a probe. And it's noisier by just technical realities. And then the sequencing, you're sequencing the RNA, so it's much more. I think it's much more test retest is you have a lower error.
So it's a transcription based test
No.
because
This was epigenetics.
no, I'm talking about the one that you prefer because we've talked on the show. I had Matt Caberline on for once and talking about how epigenetic clocks are not really accurate, or they basically from person to person or the same person might give you pretty significant differences in biological age. So it's not something necessarily that I think you might want to rely on, but You're saying there's another clock, a transcription type clock. Could you tell us how that works?
So that basically works by using blood that we lyse. So the cells, we extract the RNA from them, and then
Yeah.
we sequence all of the RNA molecules from that blood sample. That's called, RNA sequencing, which is much more accurate than the epigenetic clocks in
Mm-hmm.
our hands.
Yeah.
But the epigenetic clocks are good if you're using many and then
Mm-hmm.
you're averaging them. And so we use 35, 36 total clocks. And what we did was observe which one of these buckets of clocks, right? There's the systems age and the
Sure.
kilo age and the fitness age and the stochastic age clocks and all of those. Which ones are more affected? And then once we've done that, we averaged all of these clocks to evaluate the overall response. So when we average the clocks, I was alluding to this denoising process that ultimately you can actually compare patients before and after and have a pretty good accurate response to see if you get an improvement in this metric.
Yeah. So let me ask you this: in terms of simple lifestyle changes, dietary, pharmacologic, you mentioned some nutraceuticals. Are you seeing anything else that meaningfully shifts eye age?
um We haven't done the final analysis, but there is a study where we have exercise modalities with cycling, jogging, walking, yoga, Pilates, with zone 0, 1, 2, 3, 4, 5. We have all the zones and the strain in your, and we have all the RNA sequencing paired with these different, but we haven't
Yeah.
finalized the analysis.
Oh, okay. Well, that'll be useful to know. Help us guide some of our. Hopefully, you have high intensity interval training in there too.
It's there.
Okay, yeah, because that's the one that seems to always surprise me with
Yeah.
its effects. So these biomarkers that you guys use in terms of first of all, is eye age, is that something that's commercially available for people to
Yes.
use?
Yes.
Okay.
We have a company. It's called Edifice Health,
Okay.
and it's a company that commercializes this test. Currently offered through clinics and longevity doctors, and ready for DTC early next year. We've got CLIA approval and all the necessary regulatory approvals to go direct to consumer.
Very interesting. So, and that's going to include all these biomarkers, which we don't test right now. I mean, as physicians, I mean, I think the best for myself, pretty much, I've been relying on high sensitivity CRP, pretty much as the best way to at least get a general sense of whether I've got a lot of systemic blood vessel type inflammation. And along that lines, one thing that I'm wondering about is other pharmaceuticals. And do you think that they make a difference in terms of outcomes? So, for example, one of the drugs that some of the cardiologists are recommending now for people who have got high sensitivity CRP is colchicine. And colchicine will bring down your score, your high sensitivity CRP. But the question I've always wondered is, does bringing that number down, does that actually reflect, is that just changing a biomarker, or is that changing disease risk?
Right. Um, it's the $1 million question. We don't know. There's
Yeah.
very few studies, if any, addressing that huge gap in
Yeah.
knowledge, which is we are so happy of publishing, to publish this paper where we were able to decrease biological aging by 3 years, right? People are jumping all happy because they lower by 6 years. Whether or not that reduces risk for disease long term, we do not
Yeah.
know.
Yeah.
We assume it will, but we don't have the data.
Yeah. I guess that's kind of what I was getting at. I mean, you can kind of assume I will, especially I guess if you're using eye age and you are seeing it go down and you have various compounds in there that are causal, that makes a difference. So,
Absolutely.
right. Let me ask you I'll finish up with a sort of a bigger question. You're part of the Buck Institute, which, is really cutting edge, obviously the leader in the country when it comes to longevity research. You're surrounded by all kinds of stuff going on, from senescence to AI discovery, drug discovery research. So what's the most exciting direction you see going on right now in longevity science in the next decade?
There are, I guess, a couple of areas, not just one. I'm really excited about the possibility of identifying personalized therapeutics for different profiles. What may work for me may not be the best for you.
Yeah.
I'm not that kind of person that thinks one size fits all will work here. Okay, in terms of age optimization, right, and biological age improvement, and so working very, very actively on developing those mappings between multi-omics assessment of biological aging markers and different therapeutic interventions, right? That's one big piece. The other big piece that I would love to kind of put out there, and I'm super excited about our progress there, is the democratization of these different diagnostics. I'm developing digital markers for organ aging. And say your favorite wearable marker for organ aging, your selfie, the photo recognition as a proxy for biological and inflammatory aging. We're 90% done with this program. Is called the Healthy Selfie. And we're going to put it out there, and it's going to be completely revolutionary because you can deploy this anywhere, anywhere in the world. And then the third piece, and this is the last one, is clinical trials, right? At the moment, at the Buck Institute, we have an incredible team with Brianna Stubbs and John Newman, who are in charge of this HealthSpan Center that enables clinical trial development. And so we can recruit people, we can push the science forward, and then make the claims reality. Yeah. Because there's a lot of hype and a lot of claims that we don't know, are real. So I think there's much more research to be done.
Yeah, sure. Absolutely. Well, this has been a very fascinating conversation, and I want to thank you for being on the show today, and definitely be interested in trying out your iAge product. And I'm kind of afraid to do it, but I will anyway.
Thank you so much, Buck.
Take care.
All right, you too. Bye-bye.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.